All right, well, it is my great pleasure to introduce Nora, who I know most of you actually know because Nora's been around for a while. So she grew up in the Northeast, originally from Boston. Her dad makes beautiful coffee cups that I enjoy every morning. She went to Kenyon College for her undergraduate, where she majored in biology, and she was doing an undergraduate project with a colleague of ours, Siobhan Fennessy. And I actually met Nora after that because she was working at Nature Conservancy for a year. And so she was ironically working on sites that ended up being part of what she ended up doing for her dissertation. So I have to say that Nora is probably the most resourceful of my students that I've ever had. She arrived and I went on sabbatical, so that was like terrible of me right off the bat, right? She managed to get started and to figure things out and to work with a large group of students that we had at that point. And then she weathered COVID, and she's actually my last COVID student. So we're finishing up. We managed to get everybody else done, and Nora's the one that's been indispensable, really. She single-handedly helped me move the lab from micro back to H.J. Patterson. And done pretty much everything, and is now training kind of a new set of graduate students and helping. And I'm gonna miss her terribly. She's got another semester, and she's actually a recipient of the Anne Wiley Fellowship. And so she'll be working on that, without funding next semester. Without further ado, I'll let you get started.
Okay, thank you. That's very nice.
Just the lights in here, you know,
Are these them?
Perfect.
Thank you. Okay, well, thank you. All right, so I'm Nora Hamovit, as Stephanie said, and today I'm going to just work through some of the work that I've done as part of my dissertation research. So very generally, I study wetland microbial communities and methane biogeochemistry, which you can probably tell from this title. And hopefully I'll teach you a little bit about wetlands and restoration and methane along the way. One sec. Okay, no, that's too far. Okay, so to start, I'll give a little introduction, why I think it's important we study restoration wetlands, and why I think it's important I study specifically the wetlands I study. Then we'll work through some experiments I did using 13C DNA stable isotope probing, and also in some experiments we did across a larger number of these restoration sites, looking at methane cycling communities and methane biogeochemistry. And hopefully then I'll finish up talking about some of the next final steps we'll be taking, and that'll be sort of the flow of the talk. So to start, I think it's important to give an idea about why I study methane. Methane is a really potent greenhouse gas, and it's been rising in the atmosphere in concentration since the Industrial Revolution, but we've seen this sort of more rapid increase over the last decade or so, and this is sort of renewed interest in thinking about the sinks and the sources of methane and atmospheric methane. And so wetlands are the largest natural source of methane. This is because the water saturated soils of wetlands, they create the conditions that are necessary for the microbial communities, methanogens, that produce that methane. And so, right, wetlands figure pretty prominently into the global methane budget. On the figure here on the right, just showing that under a warming climate, we have some data that maybe even suggests that wetlands are going to start producing more methane than we'd even predicted. I say all this though, I think it's important to note that they also store a lot of carbon. So, right, wetlands, they act as natural carbon sinks. And so despite the fact that they produce methane, they're really important ecosystems. The caveat to this is when we restore wetlands, we often see an immediate return of methane production, but it takes a long time for carbon to be sequestered in soils. On top of that, when we restore wetlands, they can often show heightened methane emissions or methane that is higher or more variable than their natural counterparts. So the reason for that can be really variable and is very site dependent, but I like to use this diagram as a way to sort of think through it. If we think of restoration and past land use change as sort of a form of disturbance, We know that disturbance in a site is going to shift edaphic and environmental characteristics. So things like vegetation, soil carbon, soil nutrients, those are going to be impacted by disturbance. And in turn, that's going to affect the soil microorganisms. So think about those methanogens that I talked about that produce methane. And so, of course, if those communities are impacted, well, then microbial activity like methane production and consumption, which is just the microbial consumption of methane, that's methane consumption, those are going to be shifted as well. And ultimately, on the ecosystem scale, we're going to see a change in ecosystem functions like methane emissions, right? So we can kind of think of this general chain of why we might see a shift in functions in a restored wetland. Before I get into talking about how this sort of diagram shows up in the sites I study, I should probably just introduce the sites themselves. So the work that I've done has been centered in wetlands that we commonly call Delmarva Bays. Delmarva Bays are small depressional wetlands. They have mineral soil, which means they're not necessarily super carbon rich. Because they're depressional, they have three general hydrologic zones. They have an open water center, usually pretty void of vegetation, symbolized here by the 1. And then they have a vegetative zone, symbolized here by the 2, and a wet forested edge. These sites also experience seasonal hydrology, and so in the spring, when the growing season starts, they tend to have a dry down event, and this is especially experienced on that wet forested edge. So that wet forested edge of the wetland is going to see some fluctuation between saturation and drying. And I think this like temporal and spatial heterogeneity makes these sites really interesting, especially when thinking about the microbial community. To give a little more background, this figure here on the right comes from one of Dr. Ravenhorst's students. He used LiDAR imaging to estimate where Delmarva Bays may have once existed across the Delmarva Peninsula. So, right, as you may have guessed, if you're familiar with Maryland, a Delmarva Bay is present on the Delmarva Peninsula. And so with this LiDAR imaging, he found where depressions may have been. And so each one of these orange dots represents where Delmarva Bay might have existed. But a lot of these wetlands were lost for agricultural land use, sort of reflecting what was seen across the US over the last 200 years, right? We saw a ton of wetland land lost to agricultural and other uses. Then in the 1980s, when our sort of policies regarding wetland conservation and replacement changed, we then began to see a lot of restoration take place across the US. And sort of mimicking that again, we saw a lot of restoration of these Delmarva Bays on the Delmarva Peninsula. So my point here is that numerous restored Delmarva Bays exist across the Delmarva Peninsula. And so there are many of these sites. And while these sites provide really important habitat for things like waterfowl, I'll talk about it later, but we were at some sites this weekend, saw some cool wildlife. But even though they provide this habitat, we also know that they have these existing shifts in their environmental and their edaphic conditions. So sort of going back to this diagram, thinking of the disturbance of past land use change, and it has in fact impacted these sites. So to get more specific, a series of these sites were investigated by one of Dr. Baldwin's old graduate students. And so she looked at a pretty comprehensive number of these sites, I think about 17 natural and 14 restored, maybe the other way around. But overall, she found that these restored sites have a higher amount of herbaceous vegetation. So essentially in the natural sites, and I have this sort of visualization here, these are some of the sites that I've looked at, but essentially in the natural sites, you'll see that there's a large canopy of trees, and so it's pretty shaded out and there's not a lot of vegetation within that ponded area. Whereas if you look up at the top sort of green polygon in the restored sites that represents that vegetative zone, you can see it's relatively expanded in those restored sites. And then I have this picture here just to sort of further demonstrate how these sites are full of vegetation and sort of lack that overstory canopy. And so that's a big difference that you see very generally between these natural and restored sites. Then if you look down into the soil, there's also pretty different carbon dynamics. So these are stored sites. These are also this is data also collected from some of Dr. Rubenhorst's old students, but they have faster carbon degradation, and in turn, these restored sites have much lower soil carbon. And I included these photos here, I'm very visual, so at the top is a core from one of my restored wetlands. Hopefully you can see from the PowerPoint, but it's much more carbon poor, right? Whereas below is a core from one of my natural sites, a much more carbon rich core. On top of that, these sites are very compacted. Compaction happened during agricultural land use, and then also this contributes also to the differences in soil carbon, but the sites that I studied were all scraped, so the top layers of soil were removed during restoration, and this also contributed to compaction. On top of that, we know these sites have a higher soil pH, so they're not as acidic as their natural counterparts. So when I approached setting up these experiments with this interest in these wetlands, I knew from looking through all this literature and work that had been done in these sites that there are these existing sort of shifts in them. We also had some preliminary evidence that these sites were in fact capable of producing more methane than their natural counterparts. And so with these sort of two things together, I became really interested in understanding the microbial community of these restored Delmarva bays, and specifically understanding the microbial community and the methanogen community, and how shifts in those communities may then be evidenced in shifts in activity like methane production and methane oxidation. So when we set up our first experiment, this was sort of the general goal I had in mind, understand these microbial communities. And so to do that, what we began with is we collected intact soil cores from the wet forested edge of one restored and one natural Delmarva Bay. These Delmarva bays are the ones I refer to when I say we have some preliminary evidence that the restored site is able to produce more methane than its natural counterpart. We collected also on that wet forested edge because that's a part of the wetland that again is experiencing more variation in hydrology during those seasonal dry downs. The intact cores were collected in sterile glass tubes. There's a photo of them here. You can kind of see them, but I have sort of a diagram that maybe is easier to see. We took the cores, we put them on incubations, little incubation stands. We installed redox probes. So these redox probes allowed us to essentially monitor the level of saturation, to simplify it. And then we also had caps, and these caps were airtight. So a headspace collected above our cores, and we could then look at the headspace for things like methane, so look at methane production. The sort of most important part of these incubations was these intact cores were then fed with a heavy isotope labeled carbon substrate, specifically a 13C carbon labeled acetate substrate. We used acetate because acetate's used for like 60 to 80% of methane produced in sites like these. And it's also used by microorganisms that are going to compete or coexist with those methanogens. And so we knew essentially these communities are using acetate. I think what's more exciting though is the use of the 13C. So 13C is a stable heavy isotope, and when we fed it to these microbial communities, it allowed us to then downstream separate and identify just the active portion of the community. So to make it simple with a visualization, these microbes that were using acetate would take that 13C, incorporate it into their DNA, and then microbes that were either inactive or were just at least not using acetate, their DNA would not have 13C in it. So later, when we use DNA to identify the microorganisms, we could separately identify that active acetate using community. This is a really big beneficial sort of method because when we look at soils, they're full of DNA from dead or inactive community members. And so with typical analytical techniques, you're going to capture that whole community. But with this, we were able to look at just that active community, which gives us a much better understanding, or at least like a leg to stand on when we're trying to make assertions about the activity occurring. Lastly, these incubations, they were collected from those wetlands under a flooded condition. So when the wetlands were saturated, And half the cores from each of those wetlands remained in that flooded condition, and then half of them were slowly dried. And that was to mimic, again, that seasonal hydrology, that fluctuation that we see in those sites. In addition, there was a third group from the natural wetlands that were dried in the same fashion, and then halfway through experienced a re-wetting. And again, this just was a further understanding of how the active community may shift with hydrology. So very generally, what did we expect to see? Well, sort of just to start off, we expected that the restored flooded and restored dried communities would be distinct from the natural flooded and natural dried communities, as well as the natural dried re-wet community. So just this idea that there would be distinct active microbial communities in these incubations. More specifically though, we had some ideas about methanogens. What we expected was that methanogens would be particularly abundant in the restored flooded incubation, and that those methanogens would remain more active even when the soil underwent drying. So to explain, I'm sure a lot of you are somewhat familiar with the redox ladder. For those of you who are not, the redox ladder proposes that processes towards the top are going to occur before processes at the bottom based on energetics. And so our idea was that perhaps in these restored soils, there was either a loss of community members able to perform these processes that would outcompete methanogens, or a loss in these terminal electron acceptor availability that would also mean that these processes did not occur. And so this would allow methanogens to flourish and be highly abundant in that restored wetland under that continuously flooded condition. Essentially, they would have no competition for that acetate we were adding. We also expected that these cores that these microbial communities of methanogens would remain active in the restored core even as it was drying, because we know these cores are compacted, so we know water moves through them more slowly, and so we knew that the oxygen would also move through them more slowly, and the conditions favorable to methanogenesis would be more prolonged. And so that gave us an idea of perhaps we'd see methanogens be active for longer even as the soil dried in that restored site. We had some ideas about perhaps resistance to subsequent disturbance because of the past disturbance these communities exhibited, but we're not going to touch on that too much today. So to start, very generally what we're seeing here is an NMDS based on Brady-Curtis dissimilarity distances. Very generally, you can just say that each point represents the community composition from one of these cores, and the further away 2 points are from each other, the more different they are from each other. So the closer they are, the more similar they are. And I'm just putting this up here to show that we can see there's a pretty tight grouping of the restored wetland cores communities to one another. And that is beginning to show us that in hypothesis 1, this idea that they'll be distinct from one another is probably in fact true. And when we add in inundation treatment, we see that in fact this is true. So what we're seeing here is the natural dried wet, and the natural flooded are more distinct from one another than the restored dried and restored flooded. So also some evidence going towards this idea that perhaps the restored community either was resistant to change inherently to the community or because of the cores themselves being slow to dry. So that's sort of our general understanding of these active communities, but to get into the methanogen communities, because that's what I think is most exciting. So here we're looking at just a box plot that's showing the average absolute abundance of methane producers, so those methanogens. You can see here the average absolute abundance of methanogens is in fact highest in that restored flooded community. So this is telling us that methanogens, they were able to access that acetate that we put into those cores. In addition, the second most abundant is this restored dried community. So again, sort of pointing towards or suggesting that these community members were in fact able to remain active even during some of that soil drying. In both cases, in both wetlands, you can see that the abundance of methanogens decreased with soil drying, which again would be expected because saturated conditions are what favor methanogenesis. I think it's also interesting to point out that there's a bit of an uptick in the methanogen abundance in the natural dried re-wet core, so that core that was dried and then re-wet, and perhaps maybe a suggestion that these methanogens were not able to be active under the continuously flooded condition of the natural wetland, but were in fact able to be competitive upon initial rewetting. To understand this more though, I think it's interesting or important that we look at the composition of the community. So what we're seeing here is pretty much the same figure, but we're looking at the genres of the methanogens. And what I'll point to us specifically, because there's a lot going on here, is the pink or purple bars. You'll see Methanosarcina. Methanosarcina are a methanogen genre that are really good at using acetate. They are competitive for acetate, especially under relatively high acetate conditions. And you'll see that the average absolute abundance of these community members were really high in both the restored flooded and restored dry. So this is giving us further evidence that they were in fact able to compete for acetate and use that acetate to produce methane. And this fits with our finding that during a 2-week sort of pre-incubation under those flooded conditions, we saw the restored cores produce methane, but we saw no methane being produced by the natural cores. So to us, this indicates that there's a community composition in the restored core that's going to produce methane before the natural core. And so all together, this is sort of lending more and more evidence to the fact that there are in fact more methanogens, and specifically methanogens that use acetate to produce methane, and they are in fact producing that methane. So then this idea that perhaps this is from a lack of competition. So we looked at the abundance of microbes known to compete with those methanogens. So on the right, we're seeing the average absolute abundance of iron reducers, and over here we're seeing nitrate reducers. And the main takeaway of these two figures is just that there isn't a lack of iron or nitrate reducers in the restored cores. So basically these methanogens are competing for acetate and they're flourishing, even though there are microbial community members that are active and competing with them. And this is supported also, we looked at some iron in the soil at the end of the incubations. And so we found there was ferrous iron in these cores. We expect to see ferrous iron if iron reduction has occurred. And we also saw pretty similar levels of ferric iron, so what would be used for iron reduction, in the two cores. So basically signaling that there isn't a loss in processes like iron reduction or nitrate reduction. There's really just this difference in methane production. So when we came away from this experiment, we were left with like a really strong understanding of what these active communities look like. We have a really great data set of what these communities look like. But in regards to methanogens, I was sort of left wondering, well, is this methanogen abundance typical of these restored wetland sites, right? We had a pretty limited scope. We looked at one wetland, one like a hydrologic zone within that wetland. And so this question of, well, is this something that we see across these restored sites? Is this something we can say is typical of these restored sites? And does it correspond to a higher potential for methane production as well? So if there are methanogens there, are they able to produce more methane than the communities in the natural sites? And in addition to that, are they there even when we're not feeding them with acetate? And so again, that brings up the idea of, if so, then why? Is there truly a loss of, it's not a loss of competition, so that's what objective one seems to suggest, but is that true? Or do we see further evidence that of the role in carbon dynamics in differentiating these communities? We know that there's differences in soil carbon between these natural and restored sites. And so more specifically, what I mean by that is we know that these sites have higher herbaceous vegetation, which we know just from literature and wetlands in general, means that when soil enters the system, it is often used by acetoclastic methanogens. So there's this sort of known connection between increase in herbaceous vegetation and primary production and acetoclastic methanogenesis. And so when we approached objective 2, I was really interested in understanding, well, do we see that in fact the soil is not going to building organic matter and soil carbon, but is in fact flowing all the way to acetate and the acetoclastic methanogens? That's what I was really interested in understanding about these. So to walk you through what we did again, this time we went to 3 restored wetlands and 3 natural wetlands. All of our restored wetlands were restored in 2004 using that same soil scraping method. And we collected soil along this hydrologic gradient, so from each of these zones, the open water center, the vegetative zone, and the wet forested edge. And we took that soil back to the lab and we looked at the microbial community, specifically again focusing on methanogens. We looked at different activity, methane biogeochemistry activity. So we looked at the potential for methane production, the potential for methane oxidation, and then we looked at different forms of iron and nitrogen and carbon in these soils. So really trying to understand what's going on in these restored sites compared to the natural. So to jump right in, this is again in NMDS. So same idea here, right? The further part two dots are, the more distinct the community composition is. And I'm just putting this here to point out that to the left of this line, I'm just using to show, in blue are all the natural points, and those are from 3 different sites, and you can see they group really distinctly from the red points coming from the restored site. So even though we went to multiple different sites, they're still grouping really strongly the composition of the communities based on whether they were restored or natural. So this is just really good initial indication that we're going to see some pretty distinct features between those communities based on whether they are a natural site or a restored site. We don't have time to get into it too much today, but I will say there's also some really interesting trends between zones, and you can sort of start to see here up to the top in the squares, those are communities that come from the wet forested edge, whereas below, those are communities that are coming from that vegetative and open water zone. So that will, yeah, we don't have time to cover it, but interesting trends along the zones as well. So then, again, this is a lot. We're gonna walk through it slowly together, don't worry. But I took this composition and I was like, you know, we collected all this soil data. What's going on here? How can I figure out what's happening? And so I took all of these soil features and I, with linear regression, I correlated them to the community composition. So the length of a line is equivalent to how strongly that soil factor is correlated to the community composition. So what we'll start with first is MCRA right here. The MCRA gene is a gene that all methanogens have. And so if we can look at the number of gene copies of the MCRA gene, we can get a really good idea of how many methanogens are in a soil sample. So that's what we're showing here. This is MCRA gene copies per gram of dry weight soil. It's broken up just between each of the individual sites is grouped together. So we're just looking at the different zones between the natural and the restored types. And so we can see that very generally in the restored wetland, the MCRA gene copies are in fact higher. So this is giving us an initial idea that the methanogen community is in fact larger in the restored sites. And then when we look at composition, we are pretty struck with the fact that methanosarcina, so those methanogens that are using that acetate, are really highly abundant in our restored sites, despite the fact that we have not fed these incubations with acetate, right? So that gives us a pretty good confidence that these acetocaustic methanogens are active and present in the restored sites, even when we're not giving them exactly what they want. Okay, and this corresponds to a higher potential for methane production. So we took this soil, we set up a series of incubations to look at the potential for methane production. And what we're seeing here is the average micromoles of methane produced per gram of soil in each of those incubations. And so you can see the amount of methane produced in the restored sites is overall higher than in the natural sites. And so now we are again seeing a larger methanogen community. That community has a lot of methanosarcina that are using acetate, and they are producing more methane than in the natural counterparts. And so we collected a lot of data on different nitrogen and iron forms. We'll focus just quickly on iron. We'll look at just total iron and then ferrous iron. So a reminder, ferrous iron is what we expect to see when iron has been reduced in the soil. And so if we look, we can also see that the natural and restored site have pretty equivalent amounts of total iron. So the potential for iron reduction is relatively similar. In fact, total iron is actually higher in the restored sites. But then over here on the left, we're seeing that ferrous iron is somewhat lower in the restored sites. So this indicates that perhaps there's less iron reduction occurring, but there is still iron reduction occurring. To couple with that, we also see that there's a really high abundance of iron-reducing bacteria. Specifically here I'm showing the genus Geobacter. Geobacter contains iron-reducing bacteria that are known to also use acetate. And so together, all this data, what it indicates to me is that methanogens are flourishing and sort of they're at a high abundance, even though we have terminal electron acceptors and ion reducers present in these soils. So it's not a complete lack of competition or the ability for competitors to compete that's allowing these methanogens to do so well. So then I'm left again with thinking about the carbon dynamics in these soils. And so we also measured organic matter and dissolved organic carbon. We looked at the depth of the organic layer, and we looked again very generally at plant cover. So what we found is here showing plant coverage, so just the general percent. We found that in fact in our restored sites, matching what was previously found, plant cover is in fact higher both in the open water zone and that vegetative zone. So keeping with the trend that we thought we would see here. And this corresponds though to a lower amount of organic matter. You see on the right, the restored wetland sites have a much lower average amount of organic matter and the depth to the mineral layer. So the organic soil layer is also much lower. So this means that all this plant material is not accumulating as organic matter. And it's also not really staying in these sites as dissolved organic carbon. And so together, what this is sort of saying to me is that we know these sites have more vegetation. We know that when you have higher vegetation and you have higher primary productivity, you're going to get carbon that enters the soil. And we know that carbon is not staying in the soil, it's not staying there as organic matter. But what we do know is that there is a large acetoclastic methanogen community, and they are in fact able to produce a lot of methane. And so to me, this is suggesting this is what's happening in these sites that is allowing more methane to be produced. So the question that then sort of remained when I started thinking about these was, well, is this something that time can fix? Over time, do these restoration sites begin to store carbon and produce less methane? So can we see a building of organic matter, and can we see either a reduction in the methanogen community or a loss in the potential for methane production? And so this past weekend, we went out to 3 more restored sites. These restored sites were restored between 1986 and 1994. So if my other sites were about 20 years old, these ones are between 30 and 40 years old. And so we did the same sampling effort. We went to each of these sites. We collected soil from all the zones, the open water center, vegetative zone, and the wet forested edge. And so our next steps are again to look at that microbial community, specifically focusing on methanogens and methanotrophs, which I'll talk about in a second. And then methane activity, so methane production, methane oxidation, and forms of iron, nitrogen, and carbon. So trying to understand if over time, with more time since restoration, we see a shift in these dynamics away from methane production. So I wanted to end on maybe a more positive note, since what I've really found is that these sites produce a lot of methane. It's a lot to look at, we'll walk through. I also, for a large part of this project, I looked at methane oxidation, so the microbial consumption of methane. And I didn't have time today to focus on both, but I just wanted to sort of walk through. We looked at the PMOA gene abundance, and so the PMOA gene is similar to the MCRA gene, except it is associated with aerobic methane oxidation. And so we looked at the gene copies of that gene, and we saw that it is at equivalent levels in the restored site as it is to the natural site. In addition, there are, on the right here, we're looking at the rates of methane consumed per hour. And so the main takeaway here is that even though there are these large communities of methane producers and methanogens, we also see a return of methane consumers and methane oxidation. And I think that's just a promising note to sort of land on, is that there is sort of this natural constraint on methane emissions in these sites. And yeah, I think that's important. And so I think with that, I can say thank you to everyone, everyone who's helped and collaborated. And yeah, you can scan this for feedback. And yeah, thank you.
We have time for questions, Nora?
I have a question about your the iron data.
Mm-hmm.
And so for the iron 2 concentrations, was that like some sort of extract that you used to pull iron 2 from?
We use like a very it's a potassium chloride, like a concentrated potassium chloride extract. So it's meant to really just get like aqueous ferrous iron. And we use the Hawk like spectrophotometer and the phenolphthalein indicator.
Yeah, cool. So one idea that I have, and it would be interesting to think about this, is because you mentioned that it was the restored sites that had the highest total concentration of iron, right? And maybe that makes sense because of all that clay that's there, and there's like iron oxyhydroxides. Yeah. I could even see like splotches of it.
Totally, yeah.
But then with all of that iron oxyhydroxide that's there, and let's say that that's reflected by the total iron in the restored site, like a lot of iron 2 could actually be absorbed to the iron oxides themselves. And so it would be really cool to figure that out. Because there might actually be a lot of much more iron 2 in the restored sites compared to the natural, if you were to look at like the solid phase iron 2 versus the aqueous.
Yeah, that's a really good suggestion. We also looked, I didn't show here, but we looked at, we used like a less strong extraction to look at like poorly crystalline iron forms. Yeah. And those are, I can't remember off the top of my head, but they are, I think they're somewhat higher actually in the restored sites as well.
That makes sense.
Yeah, and so I think it'll be interesting to dive deeper into, I actually just collected this ferrocyanide data like a couple weeks ago, so I think it'll be interesting to like go deeper into it.
Yeah, and the reason I mention it too is because this is also something that's kind of interesting that I don't think anybody really knows that much about, but there can be these synergies between iron reducers, for example, in methanogens. Yeah. Where you can actually have like iron reducers like hook up with their nanowires to the methanogens and like transfer electrons. So anyways, I don't know if that's what's going on here.
No, I think that's super interesting because it's especially with methanosarcina. And so I think that like, yeah, it could be possible. It'd be really interesting. Yeah.
So I know there's a lot of ecosystem services provided by wetlands, but I mean, what you present here, especially in terms of the carbon story, do you think that wetland restoration as a strategy is still a good strategy? I mean, you certainly raised questions about the impacts of all this methane being produced by these restorations.
I think that's a good question. I think about that a lot. I think about sometimes how much methane I've produced during these experiments, just on my own. But I think sort of the first part of the answer would be, I think like some of this data is really strong evidence that like conservation is really important. Perhaps like destroying wetlands and just saying we can replace them by building a wetland or restoring a wetland is not really the best policy. But I don't think it's completely against restoration at all. I think there are tons of services, and we were just at these sites this weekend, and there's definitely carbon beginning to be built up, and like, I know it's maybe the lesser of two evils. If you're gonna have a just leave a site fallow, or if you're gonna have an agricultural site that's wet, it's probably also just gonna be producing methane. So I don't know. Yeah, I think there's hopefulness. That's maybe not.
So the big difference between the restored and the natural sites were that the natural sites were primarily forested and the restored sites were mainly herbaceous vegetation. You sampled in 3 different zones. Was there like a forested edge on the restored wetlands, and did you how progressed was it? And could you see if there was any difference between the forest and the herbaceous?
Yeah, the edges are pretty interesting. I would say the forested edge of the restored site, there's definitely a forested edge. It typically, instead of having like a overstory of trees, it'll be like small sweet gums or like woody shrubs. And so there's definitely a change in what vegetation is there, but it's just, yeah, it's not exactly the same as what's in the natural.
Kind of following on from that question, you're starting to look at older restored sites, 30 to 40 years. What are the oldest restored sites out there? And does their vegetation look different?
I would say probably there are not many sites that are older than like this 1986 site. There might be some from slightly earlier in the 80s, but that's when sort of our ideas about and policies around like restoring wetlands changed. Yeah, it's probably one of the older sites. Yeah.
How relevant do you think your results are for restored wetlands outside of the Delmar wetlands?
Yeah, so these Delmar Bay sites were part of like a larger restoration effort and then like assessment effort that was that looked at like similar sites in North Carolina, so like very generally called like coastal plain wetlands, I believe. I can't remember the name off the top of my head, but there are a lot of wetlands that are pretty similar, and so I think they also have some like connections to sites that are like just small ponded wetlands. Think of like a prairie pothole also in the Midwest. I think there are a lot of similar inland sites like these ones.
I know you said that the more fresh restored wetlands that you looked at, they use that scraping technique that caused a lot of compaction. Did they do the same thing in those older ones? And I guess you're probably, I hope, looking at like whether that compaction is starting to dissipate a little, and if that has any effect, that's kind of interesting.
Yeah, so I definitely chose the older sites to also be ones that had undergone scraping. And I will say scraping is not something we do so much anymore because I think it became relatively obvious that it's not the best way to restore. It'd be cool to look at the like compaction data now. I don't know off the top of my head, but these sites I chose these older restored sites because they were also assessed by Dr. Ravenhorst and Dr. Baldwin's students, and so they do have like older compaction data about them. It'd be interesting to know like updated if there's, like anecdotally, I'll say I was there this weekend and they are still really compacted. Yeah, they even when there's like a bit of a mucky layer that's built, you go like no more than 2 to 5 centimeters, and it's it's compacted. Yeah.
And we thought Dr. Leesner would really like the one site because there were mosquitoes.
Oh yeah, the ones we went to this past weekend were really interesting. I wonder if I have one of them was completely dried. It had definitely been really wet recently. You can't see them here, but the site, so this is Griffin here. She came to help me on Monday. But you can see this is more typical of a restored site. So it's full of like seed box, like smartweed, and then like floating duckweed, I think is the name. Not a vegetation specialist, but yeah, full of vegetation. But yeah, some of the sites that we went to, one of the sites we went to on Saturday was really, really weird, very different from the other sites.
Just maybe following up on my earlier question, there are some wetlands that were restored up in the New York part of the project, like the investigator, pretty famous wetland scientist up there, and they had data that went back maybe 55 years at the time. Interesting to look at and of course, as typical, they weren't looking very deep into the soil, 5 centimeters or something, but to get some sort of a sense of a carbon trajectory, so that, because it's hard, you're looking at something 10 or even 20 years, but if you can push that back so you can get a little bit of a line, then maybe you can do some back of the envelope calculations regarding carbon budgets on these, in terms of broad view, how much carbon is being sequestered in the soil, and what's the additional methane that might be generated based on some of your estimations. It'll be interesting to kind of see how do those fit together and how far out we have to go before we might begin to see some sort of a prevalence.
That would be really interesting. Yeah, I think I'm really excited about this data we just collected, or the soil we just collected, because I think it will be really cool to see alongside the other restored site data, and build something like
But
that.
these were the farthest back we could find in the literature in this area. But I don't,
Yeah, there was I mean, if I mention this person's name, we know him. In fact, I think Brian quoted it, cited a lot.
Kate Ballantyne?
Yeah, Kate Valentine.
Yeah, she's-
Yes.
She did a project, she worked in like the New York
Oh, in
State
those
Highway. And they were depressional wetlands,
Yeah.
and they had them that went back at that time, that paper is probably at least 10, 15 years old now, and they went back 55 years. So that,
You can read it as
it's
a,
it's further
I know.
north.
I have a-
So I mean, the
Yeah.
climate's different, so it's not gonna be exactly parallel, but
I know exactly the you're talking about. Yeah. I think actually we looked at it together one time, way back in the
In my
day.
class.
Yeah.
thank you for whatever the case is.
How many natural and restored sites did you have and where were they distributed?
Yeah. In total, so the first two sites that I looked at the first two sites I looked at are located, they're on the eastern shore, they're called Jackson Lane. And so those are one natural, one restored. I don't know why I'm going all the way back, I don't know if this is helpful. And then the subsequent next site, so the 6 sites, the natural site, one of the natural sites was the same natural site from Jackson Lane. The other restored site is a different restored site from Jackson Lane. And then there were 2 additional natural and 2 additional restored. And they're all located between I should know my Maryland rivers, but they're located sort of in the upper part of the Delmarva Peninsula. I think it's above the Chester River perhaps is what it is, yeah. And the sites we went to just recently are in the same area, but 2 of them are actually on Kent Island, which I'm interested to see if that is, you can see a difference there.
Were there methane emission measurements from these? And how did that relate to what you did?
Yeah, so the methane emissions from these 2 sites are from the flux tower data. So both of these sites have a flux tower that is using eddy covariance to look at methane emissions that are site-wide. And so I don't have the finalized version of that data, but I know that preliminary, they were seeing that this restored site was producing more methane.
You got more methanogens, but more methane oxidizers. So what's the output of that, and why are there more of these methane, metabolizing?
Like the methane oxidizers?
Both of them, why are there more? And maybe this is where it's getting at your other study, but what is it about the restored sites that makes them both sort of more productive?
Yeah, so I think what's happening in the restored sites is because we have this shift in vegetation, it's connected to this shift in the carbon entering the system, the carbon getting broken down and feeding acetoclastic methanogens. And so I think there's really a distinct community composition. So there are acetoclastic methanogens in the natural site, but they are just they're essentially, I would say, not being fed with as much acetate, and so they're not as large of a community. It's much more a balance between different methanogen genres and those acetoclastic methanogens. Sorry. Whereas in the restored site, it's just this really large community of these acetoclastic methanogens. And then I think there aren't more methanotrophs, they're just methanotrophs, so they're present in the,
They were more in the some graphs, oxidizers were more in the restored than in the natural, and also.
I think,
you had some graphs.
so
Yeah,
if
just,
you're, sorry there.
While she's looking for that, the other thing is there was evidence from some oxygen insensitive methanogens as well, right? So it seems like in the restored sites, there's methanogens that operate even when.
Yeah, even under somewhat oxid conditions.
Because in the re-wetted, there was one where you showed that it was re-wetted. You actually had higher.
Yeah,
right?
and that's from the natural wetland, and so I'm curious about that one. I think that has a lot to do with the fact that we fed them acetate, and then like when we initially re-wet them, that re-wetting had the acetate in them, and so I think that like essentially we fed acetate right to this. Yeah, so yeah, there's no like statistical difference here of methanotrophes or the PMOA gene copies at least. And you can see that like the average sort of rate of methane consumption is actually a little bit lower in the restored site. Yeah, so, yeah.
You think the herbaceous plants, something about the carbon of them is different than the leaves of woody plants? And could that have any, that just occurred to me now.
Yes, definitely. I think that's exactly it. I think like that herbaceous plant has more labile carbon inputs, and those carbon inputs are like pretty much the start of the feeding of that acetate pathway to the methanogens.
That's an interesting hypothesis.
That's my idea, yeah.
Well, that's
Pondering that interesting hypothesis.
Yeah.
I think we ought to thank Nora again for a great seminar.
Thanks, Nora.